So let me just take a moment to try to pull this all together for you and to set the stage. Because you've heard today from the first speaker about hypertrophic cardiomyopathy and dilated cardiomyopathies such as lamin A cardiomyopathy. And from Anna, you heard about the need to prevent sudden death, and sudden death is typically arrhythmic death. So I'm going to tell you a little bit about arrhythmogenic right ventricular cardiomyopathy, which is the third cardiomyopathy. And arrhythmogenic cardiomyopathy is unique in that about 90% of the presentations are arrhythmic rather than heart failure. And why is that? The right ventricle, as many of you know as adult cardiologists over the decades, has sort of been an afterthought, and most of our focus of clinical investigation has been focused on the left ventricle. And the arrhythmogenic right ventricular cardiomyopathy is a significant contributor to sudden death, which is the second largest cause of death, second behind only all cancers. Arrhythmogenic cardiomyopathy has been really difficult to diagnose, again, because it's in the right ventricle, which we haven't focused our imaging on over the years. And so up until about 1994, the people that identified arrhythmogenic cardiomyopathy were the pathologists, your cardiac pathologists and coroners, who would see the sudden deaths. One series from France identified that 11% of sudden cardiac death in patients under the age of 65 is caused by arrhythmogenic cardiomyopathy. And in children, although the sudden death rate is much, much lower in children than adults, the proportion caused by right ventricular cardiomyopathy is actually quite large. So about 25% of pediatric sudden deaths based on coroner studies in Ontario are related to either definite or probable right ventricular cardiomyopathy. So the problem in right ventricular cardiomyopathy is, at this point, is not so much how do we treat it. That's a problem as well. But the problem is really how do we diagnose it? Now, genetics help, and we have an understanding of right ventricular cardiomyopathy from a genetic point of view, in that in and around 2003, in founder populations of a form of right ventricular cardiomyopathy that was homozygous, interestingly, they also had skin disorders. And so you could identify who had the disease because they would have, kinky hair and thickened palms and feet. And so Nikos Proteinarios and McKenna and others were able to use that to identify the first gene underlying right ventricular cardiomyopathy, which is a gene of the desmosome, the structures that hold the heart muscle cells together at their end. And the gene for that one was plakoglobin. And plakoglobin is one of about 5 different proteins that form the desmosome that act as a hitch between cells, cell after cell in the heart muscle. And it was fairly easy after that to sort of use an under the lamppost approach to then look at the other cardiac genes in more heterozygous and heterogeneous ARVC to identify the rest of the genes underlying the desmosome. Now, the story doesn't stop there, because in Canada, at least, this only explains about a third of the patients or a third of the families, and the rest are gene elusive at present. So let me try to put this together for you. So we have some lessons to learn from the paradigm of skin desmosomal disease, because the skin is the other structure where desmosomes are important at mechanically linking the cells together. And so I happen to be working fairly closely on cardiomyopathy with some of our dermatologists who were noticing cardiomyopathy in their patients. And unrelated to this, but I learned a lot about skin desmosomal disease. So we came up with a hypothesis, and so I'll tell you about the testing and validation of that hypothesis, which led to a biomarker and how we identified it. So ARVC occasionally identifies isolated cases that don't seem to be occurring in families and are gene elusive. And because of our dermatology collaborations, it increased our understanding of desmosomal disease. Desmosomal skin disorders have either genetic or autoimmune etiologies. And many cases of ARVC are gene elusive, and we thought maybe some of them, perhaps just the isolated ones, are related to autoimmunity as an alternate pathophysiology, and that we might be able to find an antibody to act as a marker for the disease. But we chose to test all subjects. This work was funded by the Labatt Family Heart Center, and we felt a little spend pressure, actually, because we were getting along and the grant hadn't recruited a lot of patients. So we tested everybody, which we were going to have to do anyway as controls. And we were amazed to find a consistent autoantibody in all cases, and that's what I'm going to tell you about today. So if you think about skin disease, the skin desmosomes, desmosomal proteins are these ones: plakophilin 1, desmocollin, desmoglein 4 and 1 and 3. And the skin and the heart share common desmosomal proteins of junctional plakoglobin and desmoplakin. And these ones, when you get a double helping, give you these combination of skin and heart disease of Naxos and Carvajal disease. And then when they're in single copies as heterozygous mutations, along with the other desmosomal components of plakophilin 2, desmoglein 2, and desmocollin 2, which are in the heart, then they give autosomal dominant right ventricular cardiomyopathy. And so we knew that from working with our dermatologists that skin disorder patients also get diseases related to antibodies and not related to genes. And this is pemphigus foliaceus and pemphigus vulgaris. So we wondered if there were autoantibodies to one of these components as well. So this is an example of these hitches. So if you think about the inside of a cell and the plasma membrane and the outside of the cell, these portions of the desmosome, and the desmosome is the whole structure here, point outside the cell and extend outside the cell in the intercalated disc space. And so these are desmocollin 2 and desmoglein 2. And there's an alternate construct of desmosomal-like structure called an area composita, and this has an additional molecule called N-cadherin. All of these molecules are called cadherins, so there's N-cadherin, desmoglein 2, and desmocollin 2. And so our hypothesis decided to first just test these components as they might be involved in autoimmunity as they extend out into the intercellular space. So we selected isolated cases, familial cases, and controls. Cases were defined by ARVC task force criteria, and it's very hard to identify patients with ARVC, and this is where these task force criteria came from. So you have to do imaging. Echo's not very sensitive. Typically, you have to do MRI. You have to look at the EKGs in 3 different ways. You have to look at depolarization, repolarization, and arrhythmias. If you have biopsy or cardiac tissue, you can look at that. And then you have to do an analysis of the family history. So the sera were assessed against the 3 cadherins, which are desmosomal, desmocollin 2, desmoglein 2, and these N-cadherins of the area composita. And we first did this by a research technique, which is Western blots. And then we followed up with a more translatable clinical technique called ELISA, or enzyme-linked immunosorbent assay. We had obtained a validation cohort, and interestingly, there is a boxer dog model of this disease. It's very prevalent in the boxer dogs, which act as a natural animal model. And we also decided to assess other forms of cardiomyopathy to make sure that our test was selective. So this is our first cohort. And this is patients from our own program who had either mostly definite and then a few borderline cases based on ARVC task force criteria. And you can see in these Westerns, I'll just say look at this one here. So these are now proteins which are plated on the Western and run, and then the Western is developed with human sera first and then anti-human antibody. And so you can see that this has been plated with desmoglein 3 and desmocolin, desmoglein 2 and desmocolin 2 and N-cadherin, and that's true for all of these in our investigational cohort. And the only thing that showed up in any of them was desmoglein 2. And indeed, out of 13 out of 14 subjects in our initial cohort, they all had anti-desmoglein 2 antibodies. Except for one, this patient A8 is the mother of a patient with defined disease. She barely made it into the borderline disease category because one of her ECG parameters was just at the edge of normal. So she's a borderline case, but was negative. We ran this against an initial commercial set of controls, 12 commercial controls. One subject showed up faintly positive, but actually less than the amounts here, except for that one. And this was a 54-year-old Hispanic woman, and that's all the data we have from that commercial control sample. So whenever you make a finding like this, you need to have a validation cohort, and so we worked with the Zurich arrhythmogenic cardiomyopathy group, and they provided 25 definite patients, and you can see all of these here, and every last one of them is positive for anti-desmoglein 2 antibody. Now remember, ARVC is a heterogeneous desmosomal gene disease, so you can have many different kinds of desmosomal gene mutations causing ARVC, and it didn't matter. So many of these patients had plakophilin 2 and desmocolin and desmoplakin and other mutations causing ARVC, but they all showed up with the same anti-desmoglein 2 antibody. We did eventually test a subset against some of those other proteins. We didn't see any antibodies. And then another set of 20 controls, this time totally clear of antibodies. Now we tested other cardiomyopathies, and you've heard a lot about hypertrophic cardiomyopathy today, but we did not see any of these anti-cadherin antibodies in any of those patients. We tested general dilated cardiomyopathy patients and did not see any. So it seems to be quite specific for ARVC. And this is a little bit more data beyond our initial cohort, so now we've included both our controls and some of our patients referred who, after assessment, ended up not being considered to have ARVC, and they're very low for the biomarker. We included some possible ARVC patients. These are patients that meet half the criteria for ARVC, but are missing half the criteria. And you can see that many of them were positive, but a few were negative. And the reason we call things possible in the criteria are because we know that some of these patients aren't going to have ARVC. Amongst the borderline, you can see the one negative that we identified, and all the rest were positive. And then as you get into definite ARVC, the amount of antibody goes up, and in our validation cohort, the amount goes up. For the paper, they asked us to do an ROC curve, and this is the ROC curve. So for those of you that are familiar with ROC curve, a bad test would have a line that just goes like this, and a good test kind of has a slope and comes up over this way. And this is an almost perfect test. This is a test at this point that is 98% sensitive, 97% specific, with an area under the curve of 0.99. We tested the boxer dogs. These are, I think, 10 or 12 boxer dogs that had definite disease, and they all show up positive. We looked at 18 dogs, 2 mongrels, and 16 boxer dogs, and we selected the dogs very carefully because we wanted dogs that not just didn't have disease, but also didn't have any family history of disease, because we didn't want to contaminate it with preclinical disease that might be based on them inheriting the disorder but not having expressed it clinically yet. And so these are dogs that are all free of disease in their pedigrees in the parent dogs and in the grandparent dogs, and they are all free of the biomarker. Now, interestingly, the biomarker does seem to track with disease severity, which makes us wonder whether or not it's intrinsically involved in disease pathogenesis. So this is the PVC count. and this is the biomarker density on Western blot. And you can see there's a fairly good line. There are some outliers, but the regression had an R-squared of about 0.7. And so we decided, well, perhaps there is some effect, and one of the best tests that we had for ARVC is to take a look at gap junction functions. So although ARVC is caused by many different protein mutations, one of the common effects in basically all the tissues that we get from ARVC is that we see a reduction, a secondary reduction in gap junction function. And we have this beautiful robot that we developed in association with Yu Sun at the engineering unit at U of T. And we basically inject one cell and then watch over time as the dye spreads from cell to cell. And this molecular weight dye has to spread essentially through gap junctions, so it tells you how your gap junctions are working. This is being done in commercial iPSC cardiomyocytes. And you can see that compared to controls and normals, that with a couple of patient samples, that there's about a 20% and significant reduction in gap junction function. We tried commercial antibody directly, and there's also a significant reduction in gap junction function. So it looks like the antibody probably has some effect directly. Now, when you find an antibody, you'd like to identify the epitope or the particular portion of the protein which it's targeting. And that helps to develop an even more sensitive assay, assuming you can get more sensitive than we are now. And one of the ways to do this is to take a look at linear epitopes. And the portion of a protein to which an antibody is targeted is typically characterized by an oligopeptide of approximately 15 amino acids. And so we took these, oligopeptides as a commercially purchased library, and they're all overlapping, and they represent the portions of the protein all the way from the extracellular end to the intracellular end, and the portion in between includes what we call an extracellular anchor portion, and then these extracellular cadherin portions 1, 2, 3, and 4. Okay, and so we'll show that in the next slide. And in this slide, the antibody reactivity was highest against the extracellular anchor portion, shown here in red, and the extracellular cadherin portion, number 4, shown also in red. There was lighter reactivity with more proximal extracellular portions. And then the intracellular portion was very weakly reactive. So we've now been able to characterize the epitope. This is important for sensitivity of the test, but let's say perhaps this is involved in the pathophysiology of disease, then the extracellular epitope and then the specific epitope also helps you design some new therapies. So in conclusion, we have Desmoglin 2 antibody is sensitive and specific for ARVC. It identifies all ARVC genetic causes, plakophilin 2, desmocollin, desmoplakin, desmoglin 2, titin, and it also identified all gene negatives, which is important because if we can identify preclinical patients as well as the clinical patients with ARVC, we may be able to do some better segregation in families to identify the disease and identify new genes. And it identified the disease in canine, and we've been trying to work with our canine colleagues to develop this into a clinical test for their disease as well. The antibody identifies severity as measured by PVC count. It induces gap junction dysfunction, and the antibody may be target for immune therapies. So I'll leave it at that. I don't want to go through, and I want to make sure we leave time for questions. And these are the people that are participated in this work. In my lab, Mina Fata obtains all the clinical data and collates all that data. She works to obtain additional samples from many collaborating colleagues now. And although our initial group was only 45 subjects, we're now aiming to collect about 500 subjects. And we already have collaborators signed up to provide those samples. This guy wearing the chef apron is Diptendu, and he is truly a master chef because nobody does Westerns and ELISAs better than Diptendu. But he's not cooking the data, and so the data that you saw today is real. This is my former postdoc, and then Dana Spears has been very crucial to this collaboration working at UHN. Chile did all of the gap junction assessments in the robot, and this is Bruce Katanak, who is a mouse geneticist by training, but is retired and runs a kennel for boxer dogs in the UK, and he's a huge boxer dog enthusiast and has brought together all of those collaborations. Thanks very much.